Thank you very much for inviting me. And hi, I'm Angus. I'm currently a postdoc in Mikael's group in Lund, Sweden. But I'm going to be talking today about some of the work that we did on the main protease of SARS-CoV-2. So given that this presentation is targeted towards PhD students, that's what was in the blurb beforehand. So I'm going to try and break down a challenge like this into bite-sized chunks, and I'm going to include little mortar boards in the top corner for sticking points. So if you see one and I don't talk about it, grab me afterwards and we'll go through. So some background. SARS-CoV-2, unless you've been living under a rock, you know what's going on. Current efforts to tackle a problem like this prevent entry of the virus into the cell by preventing cell adhesion, either by inoculating people with antibodies to prevent antigen binding, or by training the host immune system to recognize. Okay, I want to stand in front of the slides and show you. So while we all know that prevention is better than a cure, there's going to be an occasion where we're going to need small molecules to act. After entry of the virus into the cell, and you guys all know this already probably, there are two polyproteins. I'm going to do something fancy on my phone. Two viral polyproteins here that you can see. You've got the tech, you can use it. And there are two proteases that function to chop up these two polyproteins. One papain-like protease that cleaves at 5 recognition sites, and one main protease, or chymotrypsin-like protease that cleaves at about a dozen sites. It's that main protease that's the focus of this study. And because of its central role in the viral replication cycle, it's a really promising drug target, and people have been working on it. There's been a slew of papers over the last couple of years. So Mpro is a 68 kilodalton homodimer, 2 × 306 residues, with a correlation time of about 27 nanoseconds, which you really need to butyrate something of that magnitude in order to see it, even at 900 megahertz, as is shown on the screen. Sticking point number 1 for PhD students: make sure that all of your backbone amides are back exchanged. Record an HN spectrum and then a DN spectrum. Make sure your peaks are all where you expect them to be, or at least close, to avoid going down an alley. Peaks in blue here, about 30 of them, didn't back exchange. And the first part of the project was finding a refolding protocol where we didn't lose all of our sample. Talk a bit more about that later. The trick was including the N-terminal solubility tag while you partly unfolded it. Anyway, the enzyme mechanism involves a cysteine, C145, and a histidine, H41. The catalytically inactive variants, C145A and H41Q, were stable for months, long enough to really do the NMR and the structural biology that we wanted to do. Okay, assignment. At the start in 2021, 2021, we were developing the assignment of this thing. Even at 900, there's quite a bit of overlap. So we developed a couple of experiments. The first was a NOESY experiment, which here for alpha 232, if we take a cross section in F3 and F4 in the TROSY reference plane, and we go to the F1, F2 TROSY plane of this NOESY experiment, we can see residues within about 6 angstroms, which is super nice for validating assignments, even potentially de novo assignments, especially when you combine it with a NOESY experiment, where 2 NOESY mixing periods, one 300 millisecond and another 50 millisecond mixing period, can allow you to determine the distance between peaks. Yeah, the cross peaks arise from the 50 millisecond NOESY mixing and the diagonal peak intensities arise from the 300 millisecond NOESY mixing. Okay, so this method shows pretty good connectivity for a helix, as you might expect. If you've done NOEs, helices light up like Christmas trees. However, this method also shows pretty good connectivity for beta sheets. So if we take Q69, we do the same. We take a cross-section in F3 and F4. In our TROSY-NOESY-TROSY experiment, we see residues highlighted in green in the figure. Similarly, for the NOESY-NOESY-TROSY experiment, there are several readily resolvable peaks to both backbone and side chain protons in an otherwise deuterated protein. So it's good for structural validation, particularly when you've got a relatively tight limit of about 6 angstroms. But that scales with NOESY mixing period. Anyway, finally, what you might not expect is it even works for disordered regions or looped regions. Here for D197, if we again take the cross section in our TROSY-NOESY-TROSY plane, we get I-1 and I+1. And we get even more information in the NOESY-NOESY-TROSY experiment. Part of the mandate for this was sparse data. This falls well within the remit of sparse data. We collected about 0.5% NUS of this 4D data cube. And it was processable on a relatively moderate Linux machine in about an hour. So it's very readily implementable in a standard process. Within a group, we found that about a day and a half was optimal for the NOESY-NOESY-TROSY experiment and the TROSY-NOESY-TROSY experiment, again, making it easily incorporable. And why do we care about this? Well, if AlphaFold is generating models, we want to validate those models. One of the typical ways that we do that is with NOE restraint data, particularly this NOESY-NOESY-TROSY experiment with a 50 and a 300 millisecond mixing period is kind of a stand-in for a NOESY buildup curve. Well, it's 2 points. You can fit an exponential to 2 points, right? Okay. Extracting structural information in solution. What are RDCs? So there are lots of lectures out there. I'm not going to repeat them. I'm going to give a very brief overview, kind of looking at the PhD students in the room here. So your protein in solution tumbles isotropically. So solution NMR, tumbling isotropically, there's no particular preference for the orientation of that molecule in solution, for example, relative to B0. This means that the dipolar coupling, say between an NH spin pair, is completely averaged out. Now that's about 22 kilohertz if it's not averaged out. However, if we introduce something into our sample, say PF1, that preferentially aligns the molecule in a given orientation relative to B0, then these dipolar couplings no longer average out. And we see a residual dipolar coupling for an NH spin pair about 20 hertz. So it is definitely worth the title residual. We can then rotate the preferred orientation of our vectors in our aligned molecule back into the lab frame using a second order Legendre polynomial and a Saupi matrix. The Saupi matrix is a 3x3 rotation matrix and requires only 5 independent parameters to rotate the whole molecule. There's 5 parameters for a whole molecule. These 5 parameters can be determined using an SVD fit, where each RDC that you measure can be used as a reporter. So if you measure 150 RDCs, because you've got 150 assigned NH vectors, you're massively overdetermined for your 5 parameters. And moreover, once you've determined your alignment tensor, each of those bond vectors report on the local structure, the quality of the local structure. You can see how this is definitely going to tie into the AlphaFold stuff. Questions over separate domains. I'm going to get to that in a sec. Now, there are several ways to induce alignment for RDC purposes, some of which are indicated in the figure there. But there have been several excellent reviews written, one by Prestegard, and Ad recently wrote one for a much more complete list of the alignment options available. For the PhD students, again, record a deuterium 1D spectrum. I imagine your lab advisors and you will probably want to do it anyway, just to make sure that you know the alignment. You know your alignment by your quadrupole splitting, your deuterium quadrupole splitting, which you would expect to be about 10 Hertz-ish. But looking at this, you want very clean peak shapes. If you don't have clean peaks, you have sample heterogeneity. If you have sample heterogeneity, your RDCs are not going to be good. And you're probably going to need to do it again. So sample prep is sometimes a sticking point. And yeah, the maximum dipolar coupling that you're going to see is going to scale with the gyromagnetic ratio. More on the next slide. How do we observe an RDC? Well, RDCs are typically observed as an augmentation of an existing J coupling. And the magnitude of the dipolar coupling relative to the NH vector is shown in the bottom right, lambda, with ratios relative to the NH. So our best case scenario for measuring this in solution is by augmenting a high J coupling value with a high lambda coupling. A simple experiment PhD students could do on Monday is, or tomorrow, whenever, you could take a TROSY, an NH TROSY with carbon decoupling, and you could just turn off the carbon pulses. Put in a DCN labeled sample, and you will get a splitting of here glycine 2. You'll see a doubling of your amide cross peaks, split by 2J HC prime in proton and 1J NC prime in nitrogen. If we take a reference experiment, isotropic here, and a modulated or anisotropic experiment, and take the difference of those two peak positions, we get J plus D. We can extract D. That is the dipolar coupling for those couplings. Contrastingly, we can use ARTSY. So instead of inducing these couplings, if we've got a large molecule or we've got a low field, we can just use intensity modulation instead. ARTSY is a method for determining RDCs for backbone amide NH vectors using intensity modulation rather than traditional J splitting. Two spectra, a reference J and an attenuated J plus D spectrum are collected in an interleaved manner. And the RDC is derived by comparison, the intensity, same peak in the two spectra. But as you can see, even with this reduced overlap, we haven't got a J splitting. It's still quite a crowded experiment. So we extended the standard 2D ARTSY to a 3D. We added a carbonyl frequency dimension. This turns ARTSY peak picking from a crowded 2D to a much sparser 3D. 3D was also collected using NUS, so sparsity again. And this drastically increased the coverage of RDCs across your protein. Now, the 2D variant of the experiment and the 3D variant of the experiment give the same RDCs. They give the same numbers. So if you've got weak peaks, you can use the 2D experiment and take overlapped peaks from your 3D. Fitting RDC data to a model. It's important to note at this point that the derivative of an RDC with respect to orientation is strongly dependent on that orientation. So if that vector is along the same direction as one of the principal axes, that alignment tensor, the derivative is 0. So you've effectively got a blind spot along these principal axes. So there are two solutions to this. Multiple alignment media. So you align at different orientations, you average it out, or you use multiple vectors within your same sample. So you've got an NH vector, you've got a HC prime, you've got a CACB, you've got a CACO, all of these vectors. Some of them are slightly less sensitive than NH, which is why NH has been used so much. Again, for PhD students, use DC. DC is actively being used developed by Yang. It's used a lot in the US, and it just works. So now, quality factor or Q, which is a bit of a black box sometimes. What is it? It's used to report the fit quality, the structure quality. For well-behaved systems like ubiquitin, you would expect a Q value of about 0.5 to 0.2. However, Q isn't entirely removed from the alignment tensor that you fit or predict. So if you're playing around with this yourself, again, PhD students, if you're playing around with it, make sure you're fitting sensible tensors or close to sensible. So moving now on to some more experimental stuff. Here we have MPRO fitting the RDC data to PDB structure 5R8T. For the included regions, we get a Q value of 0.21. So as good as ubiquitin for something that's 68 kilodaltons, which was satisfying. Hard to get to, but satisfying at the end. Now, included regions, what do I mean? That sounds a bit alien. So to avoid the deleterious effects, really, of large outliers on alignment fit, well, fitting your alignment tensor, we excluded the N and C termini because they were floppy. And we iteratively restricted the fit of two contiguous regions, which resulted in stretches K5 to L115, T199 to L272. Now, those two included regions yielded excellent agreement with X-ray and AlphaFold models. Now, you can't really do that if you have domain motion. We were talking about, do we simulate different domains in isolation before? So if we fit the three domains separately, we get an almost identical alignment tensor, suggesting there's no motion of a domain in solution relative to the crystal structure. So the crystal structure is as accurate as we're going to get on this. It's just the fact that this domain 2, this middle region, fits a lot worse. The solution data, the solution structure is likely different to the crystal structure. And also we note that the excluded regions not only fit worse, but they have a lower secondary structure content. And the inverse is true for the included regions. Now, at the time of publication, all of this stuff was done with AlphaFold 2. At the time of implementation, this was 2021, only monomeric structures could be predicted using AlphaFold 2. Whereas in solution, as we saw before, MPRO forms a C2 symmetric homodimer. Despite this, AlphaFold 2 fit extremely well for the included regions, with Q factors ranging from 0.22 to 0.25 to 0.3, depending on how much we fiddled with it. So what did we do? In each of these iterations, we restricted the access of the AlphaFold algorithm to the MSA component. And as we saw, if it doesn't have any homologs, the structure is still good, but it's not nearly as good as if it has access to template data. Yes, also we noted for the AMBER, so the final AMBER refinement step fixes bond geometries, Van der Waals clash score, and myriad of other things, resulting in a much more sensible protein. However, we observed for the included region, the Q factor went up, i.e. a worse fit, whereas for the excluded region, Q went down, i.e. a better fit. The average bond vector angle change was about 4 degrees, with 95% of the bond angles under 9 degrees. However, the distribution is quite heavy-tailed. You can see here the angle change, the logarithm of the angle change on the left versus full structure. This is our full implementation. We weren't messing with it. This was as good as we could do. And then on the right-hand side, we've got angle change versus residue number. Credit where credit's due. The inclusion of Joseph in this project drastically accelerated the AlphaFold component of it. And any speakers looking for an expert on AlphaFold and proteins, I'm sure he'd be pretty keen to talk. So how do they compare to our experimental RDCs? So if we use the included regions to fit our alignment tensor and then predict RDCs for the rest of the protein for the best full date limited and sequence limited model, we see that they agree relatively well, except for the regions in domain 2. Now, the deviation in RDC, the absolute delta D, is mirrored by a drop in PLDDT score in those regions, but perhaps not by as much as you might expect, particularly looking at the active site loop at 145. And, you know, that's the only bit enzymologists really care about, right? Now, at the time of publication, there was over 350 PDB structures available to us, which is a position you're not normally going to be in. At a range of resolutions from about 1.3 angstroms to 2.5. So we put all of them to our RDC data because of course we did, why not? So we fit our RDC data histogram in E with a superposition of all the structures in H. And we can see that the full AlphaFold 2 implementation was better than 92% of all of the crystal structures, closely followed by the date limited model and sequence limited further behind. In F, we've got crystal resolution versus Q factor. G, we've got C alpha RMSD versus Q, both showing correlations. It is also a note here that all structures are not created equal. Of the 5 structures that were generated by the full implementation, they were not all equally in agreement with the RDC data, something to bear in mind. Now, in the last few minutes of my talk, moving on to something that we've just submitted. A few months after we submitted that RDC paper, AlphaFold-Multimer was released, which is kind of what we wanted to use this whole time. And of course, the Multimer models fit equally well to the data. However, we've got a peptide binding in the active site of our protein. We could conceivably just give 2 sequences to AlphaFold-Multimer, our peptide, our substrate sequence, and our protein sequence. Let's see how close does it get. Given that I'm going to continue talking, kind of close. So on the right-hand side, we can see an AlphaFold-Multimer simulation of a short tetrapeptide, VYLQ, that binds in S1 to 4 the intended active site subsites as it was designed to. And to observe this, we implemented a neat little trick where we added an inversion pulse before a NOESY mixing period, either on resonance at 1.4 ppm or off resonance at 100 ppm. And we subtracted the TROSY spectra to select for NOEs transferred from substrate or thiol protons to our protein. This is in a pituitary background, so background contamination shouldn't be too much of an issue. This results in our final SILLY-TROSY spectra that's indicated on the left here, with NOEs from substrate in annotated gray, from thiol protons annotated red, and plotted on the right, all which effectively validate the binding pose that AlphaFold-Multimer predicted for this tetrapeptide. Note to PhD students, if your inversion pulse touches water, even by 1%, get a lot of bleed through peaks, and it really complicates your analysis. So really make sure that you got 0.0% perturbation of water. It'll speed you right up. Okay, so we had BYLQ. We also added a decapeptide. So 5 residues before cleavage, 5 residues after. For the tetrapeptide in B, D, and F, and the decapeptide in C, E, and G, we can see that all of the shift perturbations were localized to the active site, which is what we would expect. We saw intermediate exchange in F with slow exchange on the NMR timescale in G for the decapeptide. Now, mapping these CSPs, chemical shift perturbations, onto a AlphaFold-Multimer prediction of monomeric MPro bound to a decameric peptide sequence. I won't repeat it. We can see that the CSPs are entirely consistent with the binding pose that was predicted. Moreover, we can record just simple proton-proton NOESY at slightly sub-stoichiometry and demonstrate that there's no free substrate in solution, indicating a one-to-one binding, which suggests, well, it shows that both monomer active sites can be occupied simultaneously. And in J's just assignment purposes. While CSPs are good, intermolecular NOEs are even better. So in A, we've got assigned NOESY strips of the protonated substrate decapeptide that are entirely consistent with the catalytically competent binding pose that's predicted. In E, even down to the interface between the two monomers that comprises the top of the active site, as shown in F with the companion monomer and terminus shown in cyan at the top. In B, those NOEs from A are colored cyan, again, corroborating our binding pose. And one of the things, if we plot our PL-DDT score, so our confidence interval, predicted by AlphaFold-Multimer, it's relatively flat across the whole protein. The monomer drops at the N and C termini. This is the termini increase in confidence for the dimer. However, for the substrate, for the decapeptide substrate, we can see for the first 5 residues, the PL-DDT score is higher compared to the last 5 residues, which is consistent with the higher number of NOEs that we observe. PhD students in the room, as a showcase of why you should record a 3D NOESY-TROSY experiment in pretty much all of your assignment experiments. Not only do your backbone amide region turn into a barcode that's pretty easy to read and confirm your assignment, but if you have cysteines, cysteine thiol protons light up, and you can use them almost trivially to confirm an assignment here. So thiol protons that are either buried or hydrogen bonded, you'll be able to see them. And you can even extract long-range distance information like between C300 and G2 here, or across beta strands, or even corroborate ring current shifts that you might expect being packed against phenylalanine. Leaving with some pretty pictures. Qualitatively, how did AlphaFold do? So for AlphaFold-Multimer running as a monomer, on the left, we have our tetrapeptide. The binding pose is clearly quite tightly defined. Similarly for the decapeptide, the binding poses in both cases are entirely consistent with the NOE data that we observe. However, running AlphaFold as a dimer, so 2 MPro monomers plus a decapeptide or plus a tetrapeptide, we can see the clustering is slightly more problematic, and not all of these structures predicted are consistent with the NOE data. This raises a general point of maybe how much do we need to simulate? If you've got a multimer with 10 subunits that's enormous and you only care about one of them, and that's where your substrate's binding, not only will it take less time, but you might get a slightly better answer. Again, confidence for the multimer, there's a slightly larger range. But as I said before, the substrate is slightly better defined in the monomer. And with that, I'd like to say a massive thank you to Jinfer and Dusty for keeping everything running, Joseph and the rest of the BACS group for conversations, Ad for supervising the project, and now Mikhail for hosting me. And with that, any questions?